Hello everyone and welcome to today's talk in our Evolution and Ecology seminar series. Today we are very excited to have with us Dr. Adria LeBoeuf. Adria is a group leader in the Department of Biology at the University of Freiburg in Switzerland. She obtained her PhD in neuroscience and biophysics at the Rockefeller University before carrying out a postdoc at the University of Lucerne. She then did a second postdoc at the Weizmann Institute of Science before establishing her own group in Freiburg. Her current research uses social insect colonies as a model system to understand how socially exchanged fluids evolve and how they influence animal physiology and behaviour. The title of Adria's talk today is Capitalism, how the social circulatory system rewires metabolism and ageing in superorganismal colonies. As usual, we will have a live Q&A session with Adria after her talk, so please do post your questions in the designated Slack channel. So without any further delay, Adria, many thanks again for accepting our invitation to talk today, and we can't wait to hear about your work. So take it away.
Thank you so much, Sarah, and really thanks to the organizers for inviting me, for allowing me to talk to you guys about my work today, and for organizing this fantastic series. So let's get to it. So the beautiful complexity of biology around us has come about through cooperation and coordination. And at each step of increasing complexity, many individual units have come or stayed together to produce something beyond themselves. And as their genetic interests became intertwined, we've seen them essentially level up to a higher organizational unit. So these have been termed major evolutionary transitions in individuality, and we can think of them a little bit like phase transitions in biology. So generally we go from solitary individuals to a cooperative group through cooperative group formation, and then we have group transformation, which leads to a new higher level individual. So what are some of these transitions? So we have genes that have come together into genomes, simple cells, which became more complex cells with organelles inside, single-celled organisms coming together or staying together to form multicellular organisms, and even multicellular organisms staying or coming together to form multi-individual superorganisms. So each of these transitions is characterized by increasing division of labor, which is essentially a major form of cooperation. And as the level of replication changes over each of these major evolutionary transitions, priorities that were once more individualistic become collective priorities. And so essentially individual units that previously competed now cooperate towards common goals. And each of these cooperating systems must rely on an evolved set of signals and cues to coordinate their collective actions. Now, it may seem trivial that that is the case, but how coordination is actually working across some of these transitions is very tricky. And to illustrate that, let's talk about the unique biology of ants. So while each individual ant you see crawling over your kitchen counter is a multicellular organism in and of herself, the unit of reproduction in most cases is the colony, not the individual. And so here is a stereotyped ant colony. A young queen emerges from her natal nest for the first time. She mates once, and she uses the stored sperm from one mating for decades, laying millions of eggs. Now, these eggs, if fertilized, can be epigenetically guided towards either a worker or a queen fate, which is akin to soma or germline. The workers themselves fall into two somatic tissue types, nurses and foragers. And so in a worker's approximately 1 year long life, she starts as a nurse, caring for young larvae, and eventually she shifts to foraging outside the nest. And so all of these individuals in the colony, aside from the queen, are sterile workers whose only fitness is indirect fitness, achieved when the colony as a whole finally produces new sexuals. And so this is why such colonies can be considered superorganisms. So once we have our superorganism, there are many puzzles to understand how coordination works in a multi-individual system. And many of these are about collective decisions. So Collective decisions like, for example, the short-term collective decision of how do I know as a superorganism if I'm hungry? With many individuals, with their many individual bellies, how do you know when the superorganism is hungry and needs to go forage or for specific types of food? So that's one of these puzzles. Another is about coordinating for specific tasks. Like, for example, say we find a cricket outside and we want to bring it back to the nest. How do we do that? How do we get past obstacles when we don't have hierarchical control? It's only a distributed system. A more long-term collective decision is how does the superorganism know when it's time to sort of go through ant colony puberty, when it's time to go from a growing phase into a dispersing phase, from producing workers into producing new sexuals. But we also have another exciting puzzle that I call the mystery of the billionaire queens. And that's what I'll focus on in this talk. In particular, I believe that understanding how superorganisms are solving these kinds of puzzles will help us better understand major evolutionary transitions at other scales. Okay, so there are many ways to be successful at life, but generally, one should live long and reproduce a lot. But to do either of those things, we need resources. And for every bit of food that we take in and we process, and we use to build other things, there are metabolic costs. Little bits of damaged reactive oxygen species that as an ensemble can be seriously detrimental and shorten lifespan. So across the tree of life, we often see a trade-off because of this between investment in reproduction and investment in longevity. With a finite set of resources coming in, one must choose whether to invest in either maintenance, fighting against our gentle decline, or to invest resources into the next generation. And any investment in one will take from the other. Social insect queens have somehow subverted this, though, with extremely long lifespans and extremely high fertility. And so these are the billionaires of the insect world who seem to have near unlimited resources to pour into both longevity and fecundity far beyond the reach of the masses. So to begin to solve this mystery, let's look at another scale, at cooperation between cells in multicellular organisms. So our cells and our organs cooperate beyond simply performing separate tasks. They actually transmit things. So they share the products of their metabolism and even the costs of their metabolism. And consequently, some tissues, like our neurons, can live our whole lives while other cells can come and go. How does this happen? And how does this happen in our bodies? And also, how are the billionaire queens possible? Metabolic division of labor. And so today I'm going to describe a solution for the mystery of the billionaire queens, and it comes in 3 chapters. First is about nutrient transformers. The second is about molecular evidence we have for this system. And the third is about the billionaire queens and their implications. So chapter 1, the nutrient transformers. So division of labor is by definition a form of inequality. There are some individuals who perform this task and others perform a different task. They are unequal. How can the massive inequalities that we see in ant colonies, for example, between queens and workers, that come about in terms of lifespan or size or variety of things, how can these come about both on an evolutionary and mechanistic scales? So first, a bit of context. So the precursors to extant ants, bees, and wasps were parasitic wasps, and they were solitary, and they laid their eggs in or on larvae or adults of other insects. And so these larval hymenoptera consumed the living larval prey from within, typically larval prey from within, we think. And growing on this rich resource, everyone is more or less equal. Everybody has the same rich food. Everybody can eat it as fast as they can. Everyone turns into an adult. Everybody's more or less equal. So let's talk more about this rich resource of larval hemolymph. So holometabolous development is a story of transformation that goes beyond metamorphosis. As larvae grow, they transform external nutrients into storage proteins that are called hexamerins or larval serum proteins. And then during metamorphosis, these proteins and their associated lipids are disassembled and then used for metamorphosis. Then what's left is put back together into the adult fat body. And in some species and of different kinds of insects, this can be enough resources for the rest of their adult life. And so it's a pretty important resource. And so we can think about these nutrient storage proteins or hexamerins kind of as biological cache. So basically this way, larvae act as nutrient transformers, consolidating this external food into storable, easily usable material. And as they grow, they slowly become more and more significant bags of this hexamerin cache. So what does this have to do with building inequalities? So from the parasitic wasps, somehow eusociality evolved in ants, and this, I'm showing you here, a phylogeny of the approximately 14,000 species that have emerged. And there are some lineages that have lower levels of inequality and other lineages that have higher levels of inequality. And we can see this simply in terms of body size. So in these sort of lower inequality lineages, you see workers and queens look approximately the same. While in the higher inequality lineages, you see the queen and the workers are vastly different in size. And what we see in body size is also curiously mirrored in lifespan and mostly in colony size as well, with the high inequality lineages having very, very large colonies. So there have been lots of people who've talked about this in different ways. For example, I think one particularly interesting approach was looking at suggesting that building cheaper workers was an important development in this lineage towards this high levels of inequality. But generally, what we're seeing is that more primitive species are sort of more egalitarian, with less division of labor. And this is consistent somewhat with the theory about major evolutionary transitions. Yeah, I could go more into that. But here we see that these high inequality lineages have extremely entrenched division of labor. So how does this come about? So from both economics and biology around us, we can see that the ability to concentrate wealth is the key to promoting inequality. So there is evidence from both of these fields, economics and biology. And so this is essentially a feedback loop here, where the ability to concentrate wealth, this concentration is a feedback. And so how do high inequality lineages build that inequality? So looking back at the ant phylogeny, we see approximately the same grouping of across the phylogeny when we look at diet. So more primitive and egalitarian ants are mostly eating a diet of insect juice, so hemolymph of either adults or larvae, like parasitic wasps did. And larvae and adults all feed on the same material, and everyone gets the same thing. And this way, it's actually quite difficult to build inequalities. More derived ants almost all tend aphids and other hemipterans for honeydew. And this has been proposed to be key in their massive radiation. Honeydew from aphids or nectar from plants is very high in sugar, but otherwise it's not so rich of a resource. So honeydew feeding ant species need to do a lot of collection, a lot of transport, and somehow a lot of consolidation to exploit this abundant resource to essentially transform sugar into ants. So insect juice into ants, very easy. Honeydew or sugar into ants, more difficult, requires a lot more consolidation and a lot more metabolic labor, yeah? So one of the necessary steps to exploit honeydew is a behavior called trophallaxis. So trophallaxis is the ingestion by one individual of a fluid that's excreted, secreted, or regurgitated by another. And there are many different forms of trophallaxis, but these three that are sort of darkened here are the most common. And the exciting thing about trophallaxis is that it transmits not just exogenous food, but also endogenously produced material as well. So indeed, during my postdoc, I looked at what was actually being transmitted in the fluid that's passed, which I'll call trophallactic fluid. And I found many different endogenously produced materials. Using proteomics, we found many different proteins, some of which are involved in digestion, that's not a surprise, immune defense, and also growth and development. We looked at small RNAs and saw there are many different types of small RNAs. In particular, we looked at the microRNAs, and there are many. We also, by accident, saw evidence of the gut flora, which also is not a surprise that it's there. And we also found a lot of nestmate recognition cues, which are important molecules for ants who are very territorial. It's very important who is family and who is not. In addition, we even found a very important insect developmental regulator, juvenile hormone, which is normally inside an individual and regulates individual development. And here we're seeing it passed between individuals. Going even a step further, I found that when you supplemented nursing workers' trophallactic fluid with some of these components, either with juvenile hormone itself or with a pharmacological inhibitor of a juvenile hormone regulating enzyme that's also found in trophallactic fluid, we see effects on the development of the larvae they rear. So both of these juvenile hormone increasing interventions lead to more larvae reared to adulthood. So I'm not going deeply into this, but it indicates that some trophallactic fluid components impact larval development. So zooming back out again, for many social insect species where trophallaxis is the primary means of food sharing, every individual in the colony is connected through this network of fluid exchange. And so we can view trophallactic fluid as a kind of social circulatory system that's distributing nutrients and signals to each member of the colony, just as our circulatory system transmits nutrients and signals to each cell in our bodies. So this leads me to our hypothesis. The trophallaxis enables a colony economy through metabolic division of labor. And so this has a variety of predictions. So first is that in these honeydew producing ants, our primary model organism here, or honeydew feeding ants, we use Camponotus floridanus and some other species around that. So we expect that workers are nutrient transformers. And so that means they're doing that same kind of labor that we saw ancestrally in larvae, taking external nutrients and transforming them into a consolidated protein resource. Second, we expect that this cache, this transformed consolidated resource, is transmissible between individuals over the social circulatory system. And along with that is that this cache represents a kind of wealth. These proteins represent a kind of wealth where colonies that are capable of producing large individuals like new queens have more of this wealth that they can transmit. So large body sizes require wealth. And third, we expect a sort of cross-feeding to occur in colonies. So that would be sort of the definition of metabolic division of labor. Some individuals perform certain tasks metabolically, while others receive the benefits or pay the cost of these tasks. And so we have auxotrophies, as is the term more from microbiology, where some individuals rely on the work of others. And so one thing that we would expect here is that where we see certain proteins being produced is not necessarily where they are present. And then a fourth prediction is that resources can be consolidated in certain individuals, for example, in queens, or more variably in larvae, while costs will be consolidated in the workers. And so an interesting parallel of this kind of system is globalization and free market economics, where materials can be produced and transmitted over long range and passed through many hands. And in such systems, we can end up with billionaire queens. So this would allow the queen to essentially gain resources without incurring the metabolic costs that are associated with aging, potentially giving her the seemingly unlimited resources for both incredible longevity and incredible fecundity. So, is it true? So, next chapter is molecular evidence. So, do we have evidence to support this theory of ant capitalism? So to look for that, we needed to look at what was actually being transmitted between individuals over the social circulatory system in different contexts. And so we leveraged both the superorganismal and within colony scales by analyzing the proteins in trophallactic fluid samples from colonies and from single ants at different stages in their life cycles. So on the colony-wide scale, we wanted to look at young colonies, which only produce very small new workers, versus mature colonies, which are capable of producing new queens and very, very large workers in our focal species of Camponotus floridanus, which is the Florida carpenter ant. So this would allow us to test this idea of wealth accumulation in mature colonies. So we would expect to see young colonies are sort of poor and mature colonies are wealthy. Then for the single individual comparison, here we're expecting to see something like those auxotrophies or cross-feeding of prediction 3. It's reasonable expectation that nurses and foragers are the two main behavioral types within the colony, and nurses are involved in brood care, foragers go outside. Maybe they're producing different things in accordance with their roles within the colony. So Sanya Hakala, a postdoc in my group, collected trophallactic fluid from individual nurses and foragers from lab colonies. And then for the colony scale, we wanted to access the maximum dynamic range for colony wealth. But unfortunately, lab colonies for this species rarely grow to the thriving maturity that they do in the field. So with my PhD student, Marie-Pierre Mauville, we went to the field to do this, to the Florida Keys, which wasn't so bad. And we took this as an opportunity to see how trophallactic fluid changes when colonies move from the field into the lab. And then as a control comparison, we also looked at whether we could detect any differences across our population, from the more northern islands to the more southern islands. And if this is fundamental physiology we're looking at, we shouldn't really see any differences here. So, okay, so what do we see? So firstly, the different types of samples show different sort of within type variation. So field samples were more complex and diverse than lab samples. And the longer the colonies were in the lab, the less complex their exchange fluids became. Single colonies sampled multiple times showed less variation, indicating that the variability that we're seeing is both environmentally induced variation and also variation between colonies. And then we also looked at the individual ants, and whether they were nurses or foragers, they essentially had similar levels of variation, which is nice. Okay, but we wanted to look at the significantly differing proteins for each of our comparisons, but especially with all of this variable variation, every statistical approach yields slightly different results. And so to ensure that we only took the most robust hits, we used 3 different statistical methods and overlaid their results, only retaining proteins that were significant in at least 2 out of our 3 analysis methods. And so the approaches were actually very complementary, and combining them allowed us to separate the signal from the noise. And notably, as you can see down in our comparing between islands, we found no robust differences there. So that's good. Okay, so here are our significant proteins, and color indicates fold change across the 3 comparisons. And the yellow dots indicate proteins that were significant in 2 out of the 3 methods. So broadly, you can see that there's crosstalk between the comparisons, with certain proteins being hits in more than one comparison. And now I'll dive into a few of the salient results. So first, let's look at the young versus mature colony comparison. So some of the top hits for mature colonies are large nutrient storage proteins, including 3 out of the 4 possible hexamerins. So this is exactly the cache that I was talking about in holometabolous insect development. These are the proteins I mentioned that provide the means for larvae to accumulate resources and then repurpose them in metamorphosis to an organismal level form of autophagy. And so the fact that we see these in adult ants suggests that our first prediction that the workers are nutrient transformers is likely to be correct. So that's first very exciting. Thank you, prediction number one. And then finding these proteins being socially transmitted also validates prediction number two, that the cache is transmissible between individuals over the social circulatory system. And indeed, mature colonies, the ones that are capable of producing large workers and queens, are the ones that are more wealthy across the board with our 3 hexamerins and also apolipophorans, which is a similar kind of molecule. The young colonies, on the other hand, had significantly more abundant simple sugar processing enzymes, things like amylase and trehalase, suggesting that they use their nutrients as soon as they get them. And so we can view these two different approaches of young colonies and mature colonies as different kinds of strategies, metabolic strategies. So resource consolidation and investment for rich mature colonies, and spend your whole paycheck on PJ for the young colonies to get as rapid growth as we possibly can get. For our nurse versus forager comparison, again, we see the same sugar processing enzymes, this time significantly high in foragers. In nurses, a number of our hits, though, were in anti-aging proteins and proteins protective against metabolic stress. So generally, one can produce antioxidants for 2 reasons. 1 is you suffer from oxidative stress, or 2, you want to suffer less. And so these could fall into either of those possibilities. They could be about maintaining individual nurse lifespan and protecting against the metabolic costs of larval rearing, indicating that the nurses are suffering. Or the nurses are nearest to the queen in the colony's social network. And so these could be fed to the queen to protect the sort of colony's germline. They want the queen to suffer less. So we don't know yet, and we're trying to figure it out. Then to test our prediction number 3, we need to find out where within the individual and where within the colony each of these proteins is coming from. And so this is rather fresh data from postdoc Matteo Negroni. And what we see is that there are 3 major locations that the proteins in trophallactic fluid are coming from in the postpharyngeal gland, the head fat body, and the gastrofat body. And this is somewhat consistent with other social fluids like seminal fluid, that the proteins that end up there come from multiple tissues. So when we look at where we find the proteins between nurses and foragers, and where they're expressed between nurses and foragers, we see that surprisingly little stays where it is made. So over here in red, in the sort of red part of this line, this is where things are produced in foragers and remain in foragers. And in blue, the blue section down here, things that are produced in nurses that remain in nurses. And so you see nearly everything falls outside of this line. So first, over here, we see some antioxidant or anti-aging proteins that are being produced near equally between nurses and foragers, but they accumulate in nurses. We see them over on this side. And then over here, we find a few of our cache nutrient storage proteins, 2 of the hexamerins, LSP1 beta and LSP2, and then vitellogenin, another very important storage protein that is involved in insect physiology. And these proteins are produced almost exclusively in nurses, but somehow they accumulate in foragers. So altogether, this clearly demonstrates our prediction number 3, that protein presence does not equal expression. And so this is a strong evidence in favor of metabolic division of labor in ant colonies and cross-feeding. So just to go over some of the evidence I provided, what evidence do we have for ant capitalism or colony economy through trophallaxis? So we do see that workers are acting as nutrient transformers, and the expression data confirms this, what we saw in the protein data, that the adults are capable of producing these cash proteins. We see that cash is transmissible between individuals, and indeed, colonies that can make large body sizes are indeed more wealthy. We see evidence of cross-feeding in that protein production does not equal protein presence. And we have not yet tested prediction number 4, but we're working on it. So hopefully we'll be able to tell you in some time whether resources are consolidated variably in queens and larvae and costs are consolidated in workers. So so what about the implications? Can we look at how the implications of billionaire queens work, and how does division of metabolic labor solve some of the puzzles I mentioned earlier? So how does the queen avoid aging? She does so by outsourcing metabolic labor to increasingly large colonies. So this is one reason that it's probably valuable for these honeydew producer or honeydew feeder ants to have very large colonies. For one is for collection, but then for nutrient transformation. So transforming these external nutrients, you don't need a big body, you don't need a thick cuticle, you need the metabolism. You need a body to do that work. And a body to absorb those costs that the queen will hopefully not have to absorb. Then how are metabolic costs distributed between individuals? Essentially division of labor regarding who produces things and who uses them. And then how does the colony shift from rearing workers to rearing queens, that idea of ant colony puberty I mentioned earlier, transitioning from growth to dispersal? So in a way, this kind of can come for free in this system. So as the colony grows, as the colony matures, there are more nutrient transformers and more consolidated nutrients that can be unequally invested to eventually produce some extremely large individuals. Okay, so zooming back out to macroevolution, note that all of this globalized resource sharing that I've been talking about requires trophallaxis, and not all ants engage in trophallaxis. So we mostly see this giant queen worker dimorphism and large lifespan differentials in species that either feed on honeydew or whose ancestors did. So on this figure you see in this sort of turquoise, species that lineages where most everybody engages in trophallaxis. In this particular subfamily of Myrmicinae, there are some exceptions. And in red, we have species or lineages where most species do not engage in trophallaxis. And we have some interesting exceptions over here in Pomeranae. And yes, so there's one sort of exception to this association between trophallaxis and this giant queen worker dimorphism. And that is army ants, these dorylomorphs here in the middle. And so these army ants have massive colonies, they have massive queens, and they feed exclusively on other insects, often the larvae of other ants, but they do not perform trophallaxis. So this lineage might be using larvae as nutrient transformers, but mostly the larvae of other insects, or they might use cannibalism. And they might just build their inequality through behavioral segregation of resources. But they're a really tricky system to study because they build these crazy bivouacs in the rainforest, they move around, they're not sessile like most ant colonies are. So, so there are a lot of mysteries about them, but I think hopefully one day we'll get to answer some of those questions. Okay, so we often talk about social insect colonies as democracies, but from the perspective of resource allocation, we're clearly seeing something more like the inequalities that come about in unchecked capitalism. So is this evolution of increasing inequality and inevitability of a major evolutionary transition? So in 2018, Cooper and West did some modeling, and they saw that within cooperative system, natural selection tends to favor extreme specialization. And so from a certain perspective there, this makes the advanced eusociality that we see in ants take on a somewhat darker tinge. when we think about it from this capitalist perspective. So back to trophallaxis, though, the social fluid exchange is really a keyhole through which we can observe superorganismal physiology. And going beyond social insects, the system may help us better understand how multicellular organisms and their tissues communicate and share resources and costs cooperatively. So a lot of what I've just spoken to you about today is from this paper that just came out in eLife in November. So you can check it out. And then there are some new things that are not yet published that are still in the works, and I'll hopefully be able to tell you more about in some months or years. And big thanks to all the members of my lab, in particular Sanya, Marie-Pierre, and Matteo, whose work I talked about today. Big thanks to my funding agency for my funding, especially the Swiss National Science Foundation, who is supporting my lab, and to all of you for attending this talk today. And I'm looking forward to your questions.
Thank you, Adria, for that beautiful talk. It's really amazing work. Just check whether any questions are coming through. I have one to start off with. So you said that some of the casts were producing different types of peptides, and these are being transferred to other casts, and they bear different kind of metabolic costs. Is there anything known about whether they have different microbiomes to deal with these different metabolic costs and kind of enable this kind of increased output by some casts over others?
Yeah, it's an interesting question. I'm not sure about what work has been done on the microbiome between nurses and foragers, but you would expect some variation. I mean, first, most microbiome work looks at the midgut and beyond, while the social stomach is the crop. It's the foregut. It's what comes before. And you would expect that foragers will probably have a more diverse foregut because they're going and eating all sorts of weird things. Inside the nest workers, like nurses, might have a more constrained foregut microbiome. That's what I would expect, but I don't know if anybody has looked into it or seen differences between in the midgut and beyond between nurses and foragers. I'm not sure.
Just wanted to say whether it was kind of a way to kind of transmit microbes between different members of the colony. But
totally possible. Totally possible. Yeah. Another interesting microbiome related thing there is that a lot of honeydew producers have very specialized microbial partners, even endosymbionts. So especially Camponotus, the carpenter ants that I'm studying, they have a buddy, Blockmania floridanus, who is obligate for them and for their digestion, probably for nitrogen fixation, which is a key issue for species that only eat honeydew or eat primarily honeydew.
Yeah, brilliant. So another question, can your data distinguish between transfer versus protein turnover or transients?
It's a great question. So we're doing some experiments now to try to get at that using some snazzy types of proteomics, which allow us to look at specifically labeled proteins between different populations. One thing that I will say about turnover is that, so this is the gut, this is the first step of digestion. And so, they're eating things from the outside. And it's pretty amazing how rich this compartment is in the endogenous proteins. Like when you run on a gel, sort of the gut fluids of another kind of organism, you see a big smear because they're digesting stuff. But here you see the same sort of set of proteins there in the band. So I think a lot of the things that we're seeing there are there, not with intention, but they're there purposefully. They're there with a purpose to get something done. So things that are very low abundance may simply be degraded other materials.
How do these theories apply to farming ants, and do the same rules apply?
farming ants as in fungus farming, I'm guessing.
Yeah.
Yeah. So most fungus farmers, they're in the Myrmicinae, yeah. And they probably, I mean, we're doing a macroevolutionary study where we're trying to figure out when trophallaxis evolved and when it did not. And right now, our expectation is probably the ancestors of fungus farmers. So early Myrmicinae probably engaged in trophallaxis, probably. So this is still work in progress. This could change before it gets published. But this is what we're thinking right now, is that these were probably ancestrally ants that did engage in trophallaxis, and then they evolved fungus farming. And so now I think they don't engage in trophallaxis anymore, most fungus farmers. So they probably have these interactions with the fungus in some interesting and more complex way.
Yeah. Brilliant. What about other Hymenopteron species? Do these kind of theories apply to other kind of like wasps, bees, and things?
Yeah. So wasps are beautiful with regard to trophallaxis, because along the gradient of wasp sociality from solitary to highly eusocial, you see the same gradient of how much they do trophallaxis. So highly eusocial wasps do a lot of trophallaxis. And in particular, they also have a very interesting pattern with larvae. So I think probably in wasps, larvae are still the nutrient transformers. Because what you see is that the adults will take their food, they'll chew it up, they'll feed something to the larvae, and the larvae regurgitate back to the adults. And it's amazing. If you poke a wasp larva, like on its sort of chest area, it will regurgitate some liquid for you. And it's suggestive that they're probably doing the transformation. So it's a different form. Honeybees, I'm not sure. I haven't thought too much about them, but I'm guessing so the workers produce royal jelly, and royal jelly is rather similar to the components of trophylactic fluid. Yeah, I'm not sure about the cache. I have to think about it a bit more.
Is there the same sort of size differentiation as well? So larger kind of queens and more workers sort of thing?
Not as big, because also there's other constraints with flying social insects. You see a lot of similarities with termites, where you have massive queens and small workers. But yeah, with both wasps and bees, there's less of a size differential between workers and queens. And you also have the issue of yearly colonies. So most bees and wasps have yearly colonies, so they can't grow in size the same way that ants can. And you don't have the same kind of dispersal.
Yeah. Brilliant. I was also wondering, to what extent can you manipulate the trophallaxis, and can you kind of block it or kind of make them produce a cocktail of different compounds? I guess you did a little bit of that in your PhD, but I guess there are caveats for that too, probably.
Yeah, so I mean, one thing that's nice and easy is to supplement trophylactic fluid with other things. That's pretty much the most straightforward way to do any of this. We're also working on doing some RNAi type work to try to knock things down, but I can't tell you results about that yet. And yeah, CRISPR-Cas9 is really sort of a dream, but right now it's only been done in non-superorganismal ants. So that means ants where the workers are not sterile. And so in a way, you have a different relationship to cooperation. Also, not just non-superorganismal ants, but ants that don't do trophallaxis. And so I think the fact that you're capable of doing that in those types of ants and you're not capable of doing it in the superorganismal ants, I think I think there's a reason for it. But yeah, it's still a lot of tools need to be developed before we can do the kind of genetic manipulations we really want to do.
Brilliant. Okay, I think that was the final question there. So yeah, just remains to say thank you, a huge thank you, Adria, for that amazing talk. We'll be returning after Christmas, so Wednesday, January the 12th, when Professor Simone Summer from Ulm University will be talking about the anthropogenic activity, impact of anthropogenic activity on wildlife health. Make sure you keep up to date by following the EcoEvo seminars on Twitter or by joining the Slack channel. And thank you again, Adria. That was amazing. And yeah, thank you all for watching. Bye.
Thank you so much.